This is the Convergent Science Network podcast. Leading researchers in the domain of neuroscience, brain theory, and technology are interviewed by Paul Verschoor and Tony Prescott.
It's Paul Verschoor, together with Tony Prescott of the Convergent Science Network podcast of the BCBT Summer School. And today we're here with our speaker, Zoltan Molnar. And Zoltan was speaking about the evolution and the development of the neocortex. So Zoltan, why the neocortex? Why do you think that's the right target to try to understand brain evolution?
So I think this issue has been around for quite a while. And in 1664, Thomas Willis started dissecting some brains. And then he noticed that when you compare a sheep brain with a human, the biggest difference was in the cerebral cortex. Brainstem, all the other regions were similar proportions. And he noticed that the cerebral cortex was much bigger in human in proportion. And also it was more convoluted. So again, in 1664, he looked at some patients with cognitive abnormalities, learning disabilities, and epilepsy, and he noticed that the cerebral cortex had extra foldings and a different shape. So he concluded that it must be the cerebral cortex, which is the substrate of the higher cognitive functions. So I believe that Willis got it absolutely right. So I think he put his finger on this. So we have a structure which really increased during evolution, and also even minor abnormalities in the cerebral cortex can have a huge, major impact on the cognitive abilities.
Okay, but now, just from some more global perspective, before we really delve into the specifics of your work, you could argue, okay, this is maybe what they saw a few hundred years ago, given the tools they had at the time. But brain evolution is not necessarily only focusing on cortex. Cortex doesn't operate in isolation, right? There's also expansion of other structures, like, for instance, basal ganglia, as an example, your thalamus, and so on. So, do you see any critical dependencies there? Or would you, for your money, you would really emphasize neocortex as the key source of what advanced our functionality as humans?
So I think we didn't have enough detailed studies where you for instance scan in lots of different brains in 3D, and then you allocate names. So for instance, parts of basal ganglia, or cerebellum, or hippocampus, or different thalamic nuclei, higher order or sensory nuclei, and correlate it to cognitive function. And I don't think it has been done properly. So I think that would be also interesting to extend these studies not only to cortex or specific regions of the cortex, but also to other extracortical areas. We all know that, for instance, cerebellum is responsible for all sorts of other functions, cognitive, language, motor, automatisms, etc. So probably the cerebellum had to evolve as well, or its connections. Interestingly, if you look at the hippocampus, I don't think that the size varies too much, no? Do you think? If you look at different species.
In food-storing animals, it's bigger.
It's really bigger. Yeah. They have to remember where they put Sometimes I wish my hippocampus would be a bit bigger to find my keys or phone.
Right. Yeah. But now, so the other thing you emphasized at the beginning of your talk was sort of the four elements of evolution that most people got wrong, or at least you pointed to one popular video on that, right? And so if you would have to characterize brain evolution, what are then the key principles that are driving that?
So the video I showed was really just helping us to get started on discussions about the principles of evolution. Because if you think about it, we are sitting here with eyes in the front with stereo vision. We have the brain developed as it is, but it's a miracle. Do you think that you could rewind this tape of evolution a few million years and then you start again, do you think we would end up with the same brain shape or sensory networks or motor networks? That would be a very interesting test, that how efficient these networks are. No? Some people believe that you would probably end up with a similar organism. I'm not so sure.
Why not?
No. So if you look at the cognitive function or some selected functions of a bird, it can be superior to us, no? So if they would have had some selective advantages, maybe, they would dominate now, no? Or
I think you could also look at events in history and say, well, if it wasn't for instance, mass extinctions caused by, objects colliding with Earth, then mammals maybe would never have succeeded to the extent they have. If you go further back, maybe vertebrates would have been less successful. So yes,
or we wouldn't be here if a dinosaur would have sneezed while our ancestors were copulating. We would So I think evolution it's a miracle that we are here now, and we have this kind of brain and this circuitry, but probably we could have evolved into very, very different directions. And that's an interesting task for you guys, because you want to look at these computational functions and you look at the brain, and this is one possible solution to solve that problem. But you could probably solve the same problem with many other circuits, no?
Well, with Zoltan, I think, in your presentation, you made a point that might contradict you, because you're also showing that many properties or several properties are highly conserved. Like, for instance, if you look at transcription during development, there are phases in this transcription that seem to be highly conserved. Or, for instance, the structuring of a cortical sheet in terms of a supragranular layer and a subgranular layer, right? So, given that you have this conservation of certain design principles, you could also argue, well, that would suggest that if I would replay the tape of evolution from a few million years back, these conserved properties would still be there, and they'd still be, if you want, forcing a certain form of development. Of course, modulo properties of an environment, but I'm not sure if the end result would have been then radically different.
I think the reason why we conserve developmental mechanisms is because they are there already. So we can't start from scratch, very radical, changes in how we construct the brain. So we can tinker with it a little bit, but not, radically new. So just to give another example, so it's a very general principle that you generate the cerebrocortical cells in an inside first, outside last fashion. But if you have one gene missing, the reelin expressed in the first layer in the Caha-Rachis cells, and they are not secreted, you reverse this inside first, outside last gradient. You will end up with an outside first, inside last reversed order. Yet, this animal is surprisingly normal. It does have some motor dysfunction because, it reels. That's where the name is coming from. But it's probably due to the cerebellar abnormalities rather than the cortex. So what would be interesting to see, if you push this guy, reeler, mutant, would that cause, major problems in cognitive behavior? Unfortunately, in human, these are lethal very early on. Intractable epilepsy and mental retardation if you have the similar mutation. But what I'm saying is that if you just change or reverse this very conserved program, you still have some self-organizing principles, so they can still wire up. They can still process some information, maybe not as well as the normal brain, but our brain is quite plastic.
But isn't this also illustrating one of the, I think, fundamental properties that you also investigate in your research and described in great detail, is that we all talk about, let's say, conserved network elements, regulatory networks, that in turn become modulated in different ways to create a certain variability of brains. While, let's say, the core elements of these systems are actually the same, but it's the regulatory networks that are changing, and as a result, expression patterns change, right? Isn't that the key underlying feature we're looking at here?
Yes, but then all these changes at some point, they have to provide you with an advantage during selection. Mm-hmm. So you set up a network and you process information, let's say visual information, and then you have several streams to process where, what. And then you suddenly recognize that chief primate is angry with you, so you can run away and you're not selected out. So you have some advantages if you have a sensory or motor system which operates more effectively. Mm-hmm. So over millions of years, you somehow have to select the developmental programs which is producing the best possible hardware on which you can then develop these circuits. No. Mm-hmm. So this is how I see it.
Right. I think you also pointed to some of the constraints that are due to evolutionary history. I mean, the example you gave from Rudolf Raff's book, The Shape of Life, how at an early stage in development, organisms seem to converge on a similar pattern of organization, which in some sense may be a forced move. If you're going to be this kind of multi-cell creature, you have to go through this. Is it the phenotypic stage, or what it's called? And maybe there are several of those in evolution. I think yesterday in Paul's talk, he was talking about the Cambrian explosion and how all the body plans, the major body plans, emerged then, and there have been no fundamental innovation in body plans since that time. It's all been evolution within certain body plans. And I guess what we know now about these gene networks is that these are very heavily conserved from the very early days of evolution, that certain gene networks exist in all these different species, vertebrate and invertebrate, and are co-opted to do different tasks. So that must be a very strong constraint on the parts of design space for brains and bodies that evolution can explore, that possibly we're only exploring one very small area of design space. And if you replay the tape and you change some essentially accidental events that have happened, maybe we could have gone in another direction, or maybe physics just requires that some of these things have to happen the way they have happened.
Mm-hmm. No, it also would mean then, from that perspective, that if you would like to develop a completely different set of animal species, you would have to rewind up till the Cambrian explosion. Because you have to go back to the point prior to just the definition of these body plans, because that's the big constraint, right?
Perhaps even earlier, because the gene networks, even the basic components of the nervous system are there.
Exactly right.
In radial creatures before the Cambrian. Yeah. So,
what do you say about that, Zoltan?
I was thinking in more advanced stages of development, when you already have, some kind of telencephalic vesicle, and then you have subpartitioning. That's very conserved in all vertebrates and mammals. But if we go a bit further, then that will be the challenge, to identify gene networks which will make us different from others.
Mm-hmm. But now in your own research, you focus very much on cortex, and then what you emphasize very much is also this radial expansion of cortex, or the growth of cortex and the processes that would drive that, as compared to, for instance, a lizard or avian brain, right? So what are the most, if we just try to turn the question we're discussing now in a bit more data-oriented version, right? So, what are the most obvious differences between such a lizard avian brain and the mammalian brain, in which we will find this cortical sheet that you really study?
So I often have discussion with Barbara Finley about this, and she is telling me that if you plot brain size and other parameters, we have absolutely just the right size of brain as total. So other species would fit in just as well, birds included. Where we disagree is that within that scheme, I think the relative proportions of of our brain, they change radically. So the sauropsids, as I showed decided to enlarge different part of the brain than from us. We decided to, actually, we did not decide anything. We were selected out because it was advantageous to have the enlarged cortex, and the dorsoventricular ridge is not, or the derivatives of these structures, whatever they are, lateral amygdala, endopiriform nucleus, or claustrum, they are not major features of our brain. So we decided to enlarge the cortex. Now, the sauropsids decided to enlarge that bowl of cells, and they have huge circuits, as we discussed during the talk, inside that bowl. And some of them, they look very similar to the mammalian circuits. And after the talk, I think Tony's comment was a very good one. So what is the selective advantage to build the cortex like we do, mammals? And I think the major advantage is that you can produce some embryonic transient circuits, and the cortex is receiving and delivering information at the time when the elements are still constructed and still added to it. So I think this is where I see a huge advantage in having the kind of structure we have. So we have a transient platform below the cerebral cortex, and things begin to line up just as we generate other cortical cells. So I think maybe this is a big advantage.
I don't think we can push that point too far. I mean, if at an early stage in hominid evolution, our line had been wiped out, it might be today crows that are recording interviews about why their brains are better than those mammals that just scurry around. So, and I think, and it's interesting that birds and mammals have both have enlarged forebrains, but they've enlarged in different ways. And I from my understanding, the big debate is, how are those similar and different? And what I got from your talk today actually was a very interesting idea that maybe there's more flexibility in the evolution of the brain than we realize, because starting from very different underlying architectures, the avian forebrain and the mammalian forebrain, although they look very different, are perhaps converging on something similar in terms of functional architecture. And is that a fair reading, or am I exaggerating your position?
Very much so, because we talked specifically about these thalamic recipient cells, and these have very similar transcriptomic networks, and we have demonstrated that you have a significant overlap between these networks in layer 4 and also in the nidopallium in the birds, although they come from different parts. Now, I think thalamic influence can elicit all sorts of differentiation on these cells. So that would be already very interesting to see. Is it activity? Is it the pattern of activity? Or you might even deliver some amino acids, maybe transneuronally from the retina, or so. All these should be explored a bit further. So there is an influence from the thalamic fibers, and we don't know what it is exactly that you have these convergent networks. And this is just the first step of the circuit. So we don't even know where to take the next step, no? In the avian and the mammalian brain.
But now in the comparison of these forebrains, can you say something about the kinds of cell types you find in mammals and birds, and how dissimilar these are?
So all our work I presented today was on regions, dissected pieces of brain, not single cell. So I completely agree. What would be really good is to go in single cell level, and then cluster these cells according to the expressed gene, and that's one way of identifying cell types, and relate this to somatodendritic morphology, physiological properties, connectivity, and then you come up with ideas whether these evolved together or separately.
Yeah, because what you mentioned in this discussion is, in some sense, there was this idea of Harvey Carton expressed in the 60s about the equivalent circuit hypothesis. And you have tried to target that by doing, if you want, gene fingerprinting of these areas to allow you to establish whether we look at homologs or not, but between bird and mammals. So, in your opinion, at that genetic, with this genetic, if you look at these genetic markers, how similar, dissimilar do these structures look? Would you declare them being homologs and that Harvey Carton had it right, or do you think it's a bit more complicated story?
So, as I mentioned, if you compare gene expression patterns between these structures, let's say hippocampus and the striatum, you notice that you have huge overlaps in gene expression pattern, and also these networks have common elements. If you then, and also I mentioned oligodendrocytes, and then the next one was very interestingly layer 4, and parts of the nidopallium where you have the thalamic targeting. Now, just because they have similar gene expression pattern, and just in the light of that particular information, you can't say whether they are homologous or not, because we developmental biologists, we like to talk about homology when they come from the same piece of the neuroepithelium, and it's not the case. We know for sure. So this is more of a convergent gene expression pattern. Now, you could argue that, okay, so why do I say that the hippocampus is then homologous and the striatum is homologous, but not the layer 4? So you're absolutely right. Just by gene expression pattern, I can't say that that piece of neuroepithelium is homologous to the avian hippocampus. It's more of the origin, lineage, clonal relationship, and the gene expression prove that they are similar. But just by gene expression, you can't really say that these are homologous.
But your definition of homology seems to be a little bit biased there, because when we talk about homology, what we're really trying to get at, surely, is that a common ancestor had a structure, and that these two species we're looking at now have inherited that structure and adapted it from that common ancestor. That's what we really want to mean by homology. And then your target. My
definition is developmental rather than evolutionary. Okay. So that is the problem, because I can't use that kind of definition in developmental terms because they are meaningless.
Yeah, but
for us.
So homology to you isn't an evolutionary concept at all.
It's developmentally, if you talk about homology of structures, it means that they arrived from the same or progenitors from the same part, relative position of the tissue. And that's not the case for these structures. If you talk about that, yes, you had a thalamic recipient cell group, which was targeted by thalamic fibers, and they then initiated some sensory experience. Yes, they perform similar function. But would that be homology?
But let's turn it around. So, in your definition, which is a more developmental of homology, aren't you actually also importing a Trojan horse? Because you said yourself that this 19th century idea of development recapitulating evolution was not fully correct, because you see divergence in development that you will not see if you recapitulate evolution. But now, if you insist that homology must be defined as originating in the same part of the neural tube, then you seem to imply that development does recapitulate phylogeny.
No, I don't imply that, but I think this is just not the case, that these two cell groups are derived from the same part of neuroepithelium and they were re-juggled.
Yeah, but you cannot say that.
Why not? I have all the lineage information. I can track the cells. I know where they come from, where they go, and they are completely separate.
Now, let me clarify. Of course, you have freedom of speech. So you can say whatever you want. Okay? In that sense, we will respect whatever you say. But the point is that if you compare a developing bird brain and a developing mammalian brain, the timing of the developmental process will be rather different. So, for instance, the neuroepithelium in which you will find your progenitor cells might start to divide at a much later stage in the mammalian brain than it does in the avian brain. And as a result, the actual layout of the neuroepithelium will have a very different shape. So to just then use that location at that point in time as your benchmark is a sure road to failure, because the developmental program is radically different.
Yes, but that's exactly what I'm saying, that if you follow the entire course of development, so you look at these different proportions of the mammalian or avian or reptilian telencephalic vesicles, if you look at all the segments, all the neurogenesis, all the migration, all the clones you get from these, and you trace the migration of these cells, I know that this is not just stage dependent. They never, ever arrive from the same. So basically, exactly. That's why I'm so radically against what you are saying, that it might be just a timing effect. And if you look at different time, yes, they were coming from there. No. There's no evidence whatsoever that they ever arrive from the same progenitors, and they just have a different migration. So they come from in very different relative positions. We have now markers, some basic homobox genes which mark some of the segments of these, and also we have some morphological features which help us, and they are coming from different parts. And the avian brain adopted a strategy to amplify neurogenesis and change the migration of those regions, whereas the mammalian is radically different. And that's why I think that's not a big problem. I don't care whether they are homologous or not. We now sorted that they are not.
Well, you're making a strong claim here, which goes back, I think you were talking about Haeckel and his idea that very early on in embryology, we all look the same. Mm-hmm. And you're saying that there's a starting point in embryology where every vertebrate has essentially the same embryo. And from that point, you can track what happens to each bit of the embryo as it moves around into
what
the
I'm
body.
saying is that the telencephalic vesicle. So let's say I give you 2 or 3 sections, matching stages from a turtle, chick, or a mouse, and I stain it with the same 4 representative homobox genes, and I cover slip it, give you a microscope, and then I ask you, okay, tell me which one is which. You couldn't be able to tell me at an early stage. So in that sense, yes, Haeckel was right that these early stages are highly conserved. But, they don't go through the, later, or especially the adult developmental stages, to go to the next stage. That's where we disagree.
But it seems a bit circular here, because you're now using gene expression to say, these are, this is my standard. This is the common pattern across all these creatures. And I know it because of the gene expression.
Mm-hmm. But you can use, Tony, you can use many other things.
Okay.
Gene expression is just one. But, people usually accept, gene expression quite well, because, these are highly conserved. You never have a Hox gene, changing its gene expression at these early stages.
Yeah, but you're now saying when we come to the forebrain, mm-hmm. that gene expression seems to suggest that part of the dorsal ventricular ridge is similarly organized to neocortex, but you don't want to accept homology. So
the gene expression not supporting that. So in fact, if you look at these these are very early studies from several groups, but basically if you look at these, there you have a couple of key genes which mark a cortex, for instance. So that's the EMX genes. Then you go to the this intermediate part, that's the PAX6. Then you have the DLX genes and so on. So the order of these and the segments, they are highly conserved early on. I'm not saying that's the only criteria. You can use many other things probably. But this is clearly indicating that, unfortunately, DVR and cortex, developmentally, they come from different parts of the brain.
But they converge to have the same pattern of gene expression in
exactly.
Adults.
So finally, But and that was a very interesting, and that's why I mentioned it in the talk. That's why it was the thorniest problem of evolutionary biology, because the gene expression, homology, and physiological properties, they all supported that, they are homologous. But the developmental data is clear that they are not. But this is just as interesting.
Be more flexible about what we mean by homology.
Yeah, I think that's the problem.
Because you're saying that at an early stage in the embryo, all embryos are essentially the same, and then you can see what goes on from there. But what we're saying now is that in the adult, although these have very different developmental paths, they've converged on a very similar structure, at least in terms of gene expression. So you could say that in the adult, they are homologous.
Right. But in my talk, I mentioned that when I use similar terms, then I was warned by others, including Neuwenhuis, who is an expert in this, that he said, you should stick to one meaning of homology, and developmentally, they are not. So that...
Okay, so Tony, I'm afraid we will not convince Zoltan of this during this podcast, but we will twist his arm afterwards.
It took 60 years for Harvey,
No, but look, so we just so homology. At least I think this discussion makes clear it's actually not completely resolved what we mean with it exactly. There's some controversy around its definition, dependent on the field in which it is used.
Exactly. Especially your field has a problem because we know exactly.
Okay. Now this is exactly what we're talking to you, Zoltan, because you know and we don't, you see. No, but the point is that then if you talk about this this sort of equivalent circuit hypothesis now, okay? So the idea would be that functionally this avian forebrain compared to a mammalian forebrain functionally might do similar things. It might show even, as you showed, right, similar response properties to visual stimuli. But in terms of its internal organization, it's different because in terms of its layering, it's different. So how should I interpret that? Because so in your talk, you showed that in cortex, mammalian cortex, you would exploit more the 6 layers of a mammalian cortex to perform certain operations. While in this avian brain, you might use a more, at least in this conceptualization also of Harvey Carton and others, a more, let's say, a sequential operation between different subzones, as opposed to exploiting parallel processing within layers. So how should I see then exactly this equivalence between these two anatomical structures?
So Harvey in the 60s suggested that if we just look at these circuits, the thalamic targeting, so the cells are in different arrangements. So the thalamic targeting cells are equivalent, let's say. Then the next step is the supragranular layers. That would be the next step. So they are located somewhere else. And then the final output layer is in the dorsal part of the dorsal cortex, hyperpolyum in sauropsids. And then you would have that in layer 5 and 6 in the mammalian cortex. So if I mentioned 2 studies, one from Suzuki, from Hirata's lab, and another one from Jennifer Dugas-Ford from Rexdale's lab, when they looked at a handful of gene expressions with the most interesting markers for the supragranular and infragranular cells, and that further confirmed this kind of segregation of circuit elements, and they supported Harvey's original idea.
But now explain to me, there's something I don't understand. If you look at the mammalian cortex, you would have a continuous interaction throughout with thalamus, for instance, right? But now in this avian solution, you would only have a subzone that is sensitive to thalamic input. So do you also see a patchy projection from a thalamus into that area? Because that's what it would imply.
So I am not an expert, unfortunately, in the thalamic projections in sauropsids, but they do have a parallel processing. No, they have so you have the collicular input is going to the nucleus rotundus, and then the nucleus rotundus will project then to a different part of the brain, to the dorsal cortex, rather than to DVR. Whereas the specific sensory nuclei, they go straight to the DVR. So in mammals, of course, you have the first order and higher order thalamic nuclei, and they have completely different layer-specific innervation the first order target mostly layer 4, although they go to all other layers, whereas the higher order thalamic nuclei, they project mostly to layer 1. And also the output from the cortex itself is different to these different thalamic nuclei. So from the primary sensory areas, you have layer 6 projection back to these, whereas to the higher order thalamic nuclei, most of the cortical input is coming from layer 5. So if you have a little bit of mismatch between 6 and 5, then you can actually communicate cortical information via the thalamus. Mm-hmm. And that's an idea which Ray and Murray Sherman developed, and it's very powerful. And that's also indicating that you have to coordinate cortical development and evolution with the thalamus. Mm-hmm. And this is not really followed up in comparative sense. Mm-hmm. So that would be interesting to see. Okay. And I think that's what you asked.
Yeah, that's a testable hypothesis then, right? That would be a consequence of this equivalent hypothesis. Okay.
But you're suggesting in the talk that DVR neurons were having some very similar functional properties to, for instance, visual cortex neurons.
Electrophysiologically, Paul Menger recorded from the iguana dorsoventricular ridge, and it was interesting to see that the receptive field properties were very similar to the mammalian primary visual cortex, and also you had multiple representations within the DVR with mirror reversals, which is also very interesting. So even if it's a nuclear representation, you still have multiple, which is also a feature of cortical visual fields. Now you have dozens of visual areas with multiple representations.
And is there something like a DVR microcircuit, which might be similar to a cortical microcircuit?
Yeah, really pushing me here. but according to Harvey Carton, yes. So he can see lots of
similar. But what's that confirmed?
Lots of similar elements, and I think this would be a really good time to tackle this in more detail. Mm-hmm. I think these comparative circuit analysis. Mm-hmm. Yeah.
But now the other thing in this comparison between a mammalian brain and an avian brain, you also looked at the development of these structures, right? And the point you were making is that the migration patterns during development to build a cortex or to build this DVR structure are actually rather different. But so what are the similarities between these migration patterns of the cells that form these areas, and what are the main differences?
So I don't know too much about this corner of the brain, where the DVR is coming from. I told you that this is one of the most complex regions. So you have a lateral stream of migrations, and then some authors like Luis Puelles, who is an expert here, is suggesting that as a field, they produce the claustrum, amygdala, and endopiriform nucleus, these regions as a field, and then they differentiate further from this stream. Whereas in sauropsids, you developed some developmental migration patterns or lack of it, and then these cells protrude into the lateral ventricle, and they form this dorsoventricular ridge. Unfortunately, I don't know too much about the subdivision with this dorsoventricular ridge, but that could be also very interesting to see how they differentiate. Mm-hmm. So this is a key feature which is different. Now, the dorsal cortex, as we discussed and argued, it's coming from a different part of the neuroepithelium, and it has an inside first, outside last pattern. And we also mentioned reelin expression, which is actually setting up this major polarity. So the inside first, outside last.
But now you did mention that if you take a mammal of which you knock out PAC5,
PAC6, yeah.
Or PAC6, sorry. Then you get in the end a brain that seems to follow this idea of a ball of cells being a forebrain. So would you then see that as an indication that to go from DVR to a cortex really depends on a single regulatory gene?
So in the talk, I mentioned a study which was done in collaboration with Anastasia Stoikova in Göttingen. And what we noticed was that if you, in the PAX6 knockout, you will have problems with this lateral stream of migration and differentiation, especially these areas, like lateral amygdala, claustrum, endoperipheral nucleus, and you will begin to see a ball of cells protruding into the lateral ventricle, very similar to what you see in the turtle. Now, we also discussed during the talk that unfortunately PAX6 is a master gene, so it's involved in lots of different other developmental steps, including cortical development. But nevertheless, this clearly indicates that, just by changing some aspects of mammalian development, you can actually regress to producing this ball of cells. So I also emphasized during the talk that it's probably not the main. I'm not saying that PAX6 was responsible for this. It could have been, but I think there was a very interesting rearrangement of that zone during evolution, and it's still reflected now in these basic developmental patterns in mammal.
But in the PAX6 knockout mice, what's the behavioral signature of that?
If you knock out in all these structures, it's death. So if you have conditional knockout, you can have cortex-specific, or you can have other conditional knockout, then it's a bit more subtle. And what is also interesting is if you change PAX6 expression at these zones, then this intermediate island of cells is blocking off the thalamic fibers of entering to the cortex. So that's why I was so interested in this mutant, because you have a thalamic phenotype. Mm-hmm. They can't make it through them, this part.
Right. So is it the case that the dorsal cortex in turtles is a true homologue in the sense that you want to use that word of the neocortex in mammals?
So the same piece of neuroepithelium is giving rise to the dorsal cortex in turtle and to the dorsal cortex of mammal, but the mammalian dorsal cortex, that part of the neuroepithelium, was turbocharged, and it's now producing lots of progenitors, and it has an enhanced neurogenic capacity.
And this may be because of genes, including PAX6 perhaps, which are involved in suppressing the development of the dorsal ventricular ridge, which otherwise would take on this perhaps higher cognitive function. And is it the case that in birds they have this dorsal cortex, but it hasn't really changed much from the turtle?
That's a very interesting thought, that to be able to promote the dorsal cortex, you have to suppress the DDR, which is very interesting. I always thought about it that you just cranked up the neurogenesis in the dorsal cortex, and then you start of sensory modalities and other motor functions were colonizing it. But your suggestion that you also have to suppress the other, then it's an interesting one. It is a possibility. Mm-hmm. But whatever happened, what I wanted to say at the middle or last part of my talk is that that part of neuroepithelium started to generate more neurons. The developmental program introduced more intermediate progenitors, and probably some of these intermediate progenitors, they are also amenable to regulation, increasing the potential that you produce neurons according to the need of the dorsal cortex. So it's another aspect of self-regulation. So it's always, unfortunately, if you block thalamic input at these early stages of development, you get the same, very similar cortical numbers. Mm-hmm. So it's not the thalamic input. It's probably there's an innate, inherent program which is producing these cell types, and the thalamic input is probably just modulating it.
But now, can you tell me, what do we know really about PAX6? What's it regulating exactly?
I mean, it's hundreds of genes. Okay. So it's very, right. So when you ask a developmental neurobiologist, okay, tell me how this transcription factor is acting. So what they will usually say is, okay, let's get some of these transcription factors, produce some antibodies against them, do some chromatin immunoprecipitation. When you precipitate these DNA parts, smaller fragments, then you pull out the parts which were specifically binding by this transcription factor. You sequence them, or you identify the genes where they bind. You have hundreds of binding partners, and then you pick one of them, and if you're lucky, yeah, that's important. So this is how the field is looking at. PAC6 has been studied by many outstanding groups, and they are slowly beginning to understand how it's regulating eye development, brain development.
But another interpretation, which relates to some other data you showed, is that actually, there's a very specific temporal patterning in the migration of these cells, and also the construction of their interconnectivity, and that in itself can form barriers for the migration of other cells, right? So to actually go from a ball of cells more in the center to a sheet at the outside, you must cross these kinds of barriers. So could the key regulatory switch be the one that's like a traffic cop, helping you to regulate the crossing of multiple migrating populations of cells through these kinds of bottlenecks? Do you see that as a possible mechanism, or is that irrelevant?
So it's a very interesting question you just rose. So this corner of the brain where you have either the dorsoventricular ridge in sauropsids, or you have this lateral stream in mammals, it's interesting that you have to clear that part by the time thalamic projections pass that region, and also corticofugals. Without clearing that in time, delaying the development or altering that stream will have serious implications on the thalamocortical targeting. So lots of thalamic fibers fail to pass through that region if you have any migration problems of these cells out of the way. So Sonia Garel in École Normale in Paris looked at some of these evolutionary steps, and basically she identified some of the factors which would help the thalamic fibers to go through the region. And there are some evolutionarily conserved mechanisms. So probably it's not a surprise why we have all these developmental phenotypes in that particular region, because it's so vulnerable for timing and migration and cell patterning.
So, but then,
you mentioned that the neocortex becomes turbocharged in mammals, but
it's a hypothesis. Yeah,
it doesn't happen just all in one shot, does it? There's stages to that. So you mentioned that if you look at an animal like Monodelphis domestica, which seems to be similar to, in some ways, to an early mammal, the density of neurons and the organization of the neocortex is much less than in a mouse, say. So, and it's got something like the sixth-layer cortex, but there are changes then that happen between that and later mammals, which make it even more turbocharged. I mean, is that right? Do you see two stages here in neocortex, or more than two stages in the way that neocortex has evolved?
Tony, this is exactly how I imagine this. I think we could argue whether these intermediate progenitors are specifically mammalian or not. If you talk to George Streeter, he would argue that these intermediate progenitors are present in larger avian brains, and they are responsible for producing more cells in the avian brain. Where we slightly disagree is that whether in the hyperpallium, the dorsal cortex equivalent part of the neuroepithelium, whether you have these intermediate progenitors in large number in birds or not. I would like to suggest that you don't have many of these there, but you might have some in the subpallium. And I think so in all mammals studied so far, including Monodelphis and wallaby, as I showed you, we can see these intermediate progenitor cells, but much later in development. And I also showed you the cell counts for Monodelphis, and it's roughly half of the mouse in a unit column area of the of the brain. And I also showed you that the onset of these subventricular proliferative profiles in the subventricular zone, they are delayed considerably, but you still have them. We have a little bit of argument whether these are neurogenic divisions or not, or they are already gliogenic with Antonello Malamaggi in the Trieste. But we all agree that eventually you will have some divisions there, and they begin to produce neurons or glia. In my opinion, they start producing a little bit of neurons as well. But we have to do some clonal analysis to be sure of that. Now, if you look at then the elaboration of these subventricular zones, Henry Kennedy will be speaking here soon. He will probably touch on this, that if you look at the primate ventricular, subventricular zone, you have several subzones with lots of other progenitors, and they start to produce, there is an explosion in numbers. And these progenitors, they produce neurons in many, many different levels. And I've shown you these interkinetic nuclear migration profiles. So they have to descend and divide there. These intermediate progenitors, they either don't have to move, like the TBR2 positive, or they move to the opposite direction. So you have 3 additional floors where you can produce neurons, and suddenly you can have this explosion of neuronal production in the brain, which is necessary, I think, to produce. And coupled with this, you have a stable platform in the subplate where you can start already building the connections. I think we have a very powerful developmental program which can not only continue to produce neurons, but already start wiring them up.
So just to be clear, the monodelphis has all the same progenitor types, or is it missing some types that other mammals have?
The proportions are different.
Right.
But in my opinion, you have the radioglia progenitor, and you also have the TBR2 positive intermediate progenitors. But these progenitors are in smaller number. Whether they have these outer radioglia progenitors or not in very low numbers, I don't know. Even in mouse, it's less than 5% of the progenitors, which is actually a very prominent feature of the primate brain. Henry will show this. Yeah.
But now with these intermediate progenitors, does it mean some sort of modular construction process during development? That means that you first can produce a small set of layers, if you want, of a cortical sheet, but then you have to inject an intermediate progenitor on top of those layers to build your next layer, etc. Is that how you see it?
I see it more of a amplification machinery. So I believe that these intermediate progenitors, they contribute to all layers, as I showed you with this Navneet Vashistha and Fernando Garcia Moreno paper, where they trace the origin of these cells. And I think we concluded that 25 to 50% of the mouse brain cells come through these progenitors. So, but I don't think similar lineage analysis has been done for the outer radioglia cells, and that will be very important, and also for the short radioglia progenitors. So I think this has to be done in the future.
Yeah, so this lineage analysis you did by actually tagging different cells with different colors in the end, right? And then you could see, okay, which cells are clones of which progenitor cells. So, but what are the key observations in that experiment, which looks pretty amazing, actually?
So we only have these experiments for the TBR2 positive intermediate progenitors because we had access to these TBR2 Cree progenitors, and then we use the clone method on the top of that. And what we could now tell is what is the average clone size, how many cells you have, how many times they divide, we are now sure that they divide at least 2, 3, maybe even 4 times. But we haven't got the data for all the other progenitors. And you will hear from Henry in about 2 days about their observations where they imaged cortical progenitors in the primate brain in vitro, and then they followed these cells to the cortex, and then they identified the phenotype and laminar position of these cells. So they could have some snapshots. They could put these primate brain slices in the dish for weeks. And then they followed them, how they divide, where they migrate. And what is interesting, and what Henry and Colette are proposing, that these progenitors, they transform into one another in different directions. If this is the case, then things will become very, very complicated, because if you can transfer TBR2 positive intermediate progenitor back into a radioglia, or you can transfer them to an outer radioglia and vice versa, then things will become very, very complicated indeed. Yeah.
But do you find that a reasonable interpretation? Isn't it?
It is, yeah. It is. But I haven't done any time-lapse studies as such.
Right. Tony, you have... Okay, so...
I mean, somebody has to listen to this podcast, and probably they are all asleep by now.
No, don't worry.
No, we're just going to do the best bit.
No, we're recording. So you in the end summarized the main results you're presenting in terms of innovation, conservation, and convergence.
Yeah.
So what did you mean with that?
So I have to come back to this topic where we spent quite a bit of time in discussing that. So after all this transcriptomic analysis with Grant Bellegarde and Chris Ponting, combining it with the cell lineage and clonal analysis and gene expression, earlier gene expression, developmental gene expression, I had to realize that some parts of the brain where you have different developmental origin, they can adopt similar gene expression. So that's what I mean by they converged to that particular. In the striatum hippocampus oligodendrocytes, we emphasize the conservation of gene expression networks. And what was the third?
Innovation.
So when you compare brain regions, that's the interesting bit. What is new? What are the mechanisms which, you don't see in other species? And especially during development, which we didn't do, that would be very interesting to see what is it? How did we get this turbocharge of the cortex? I think that would be a very interesting.
So Zoltan, you're in this business now for a while. You have gained this incredible insight in the development of the neocortex, even though we still have to explain to you about homology, but that's not a discussion. But now, given your experience in neuroscience and the study of the brain, right? If we would like to follow in your footsteps, what's Zoltan's law that we should adhere to? So Zoltan's law of the study of the brain.
So here on this course, I emphasized the comparative evolutionary developmental aspects, but most of the work I'm doing is understanding how mammalian cortical circuits are put together. And I did not really talk about this part, but I'm very interested in this early transient platform, which is below the developing cortex, the subplate neurons. So these are largely transient cells. They are the earliest generated cells in our brain. If you look at subplate in the primate brain, Andrew will probably show you some, it's bigger than the cortical plate early on during development. And then these cells, after they set up all the connectivity, integrate into the intra and extracortical circuits, then they will disappear. And you only have a couple of scattered interstitial white matter cells left over. They are cleared away, and then the final product, the cortex, will remain there. So I'm fascinated with the association of these scattered residual cells to cognitive abnormalities. So it's like when you have sloppy builders, they leave some of the scaffold behind when the adult structure is finished. And probably they haven't done a good job anyway in the adult structure because they left the scaffold inside and then they can't remove it. So I'm fascinated with these. And that's why I'm so interested in development, because I agree with Willis from 1664 that many of the cognitive disorders, they are brain developmental abnormalities, and they are related to cortex. And these transient scaffold cells, or the remnants of them, they tell us quite a bit about how the brain is constructed. And simply because these are the earliest generated cells, to understand them and understand their evolutionary origin, that's why I do quite a bit of comparative work. So although it's a bit more clinically driven, what I want to emphasize is that sometimes it's good to just step back and look at the bigger overall picture. And although it's a simplification, but the subplate is maybe the reptilian framework of our mammalian brain.
And then if we're going to meet up with you in Oxford 4 years from now, and we're going to remind you of this discussion of today, and we're going to tell you, look, 4 years ago you made this prediction, and now we're going to go check to see if it's came out. What's this one prediction you would like to make you feel most passionate about today in your own work?
So if in the next 4 years I could monitor and modulate these transient scaffold cells in the mammalian brain and show that by modulating their function could alter cognitive development, then I would be very, very happy. And if I could extend it some neuropathology, so histology or even imaging, now we have 7 Tesla machines, and if I could show that abnormal cortical development will have an impact on these subplate cells or vice versa, and then I would be very happy.
Okay, great. Zoltan Molnar, thank you very much for this conversation.
Thank you, Paul, and thank you, Tony.
The CSN podcast was produced by the Convergent Science Network of Biomimetics and Biohybrid Systems, a project funded by the European 7th Research Framework Programme. For more interviews, recorded lectures, or upcoming conferences in the field of biomimetics and biohybrid systems, go to csnnetwork.eu. And thank you for listening.